Hello, everybody. If you would, hello, is the mic on? We're going to go ahead and get started since it's 4:45. I know we'll have people joining us later. But I do want to welcome you on behalf of the Wound Healing Foundation and my co-moderator, 2016 Wound Healing Foundation Fellow, Dr. Joshua Tam, and our 3M representative, Dr. Matthew Cooper. They'll be helping me co-moderate today. And I'd like to welcome you to the 19th anniversary of our Wound Healing Foundation 3M Fellowship Award. Here are some hashtags in case you do social media, but also please note that we have the website as well as various social media. Zach, it's not moving forward. I'm on the slides, but it's not. Oh, okay, there we go. Fantastic, thank you. So please feel free to look us up, tag us, follow us, like us, just no hate mail, please. I'd also like to thank the Society and the organizers for this opportunity to bring the fellowship lecture to you during our sessions. This will be the 18th annual of the lectureship itself and the 19th Fellowship Award. And a little bit about the fellowship. A lot of people think it's just about the $15,000 fellowship grant itself, but there's actually a lot more to it. As you can see, we also now have where the Hunt lecturer, this year was Dr. Elaine Fuchs, came in and she actually had lunch with our fellows. So this is an added benefit because it's a very close mentoring and networking opportunity for our fellows to have some one-on-one time with her. And we also try to keep it involved so that one year the person receives the fellowship award, the next person that had received it the year prior is giving their result, and the person that had received it the year prior is actually moderating, so that we get a nice consecutive, helps encourage them to stay in wound healing, helps you know who some of the up-and-coming scientists and researchers are. And we also have encouraged them to publish in the society journals, Advances in Wound Care and Wound Repair and Regeneration. And keep your eye out, there's actually going to be a Wound Healing Foundation fellowship issue in Advances in Wound Care coming soon. This is a nice list of our fellows from past years. You will recognize many of the current society leadership, but this is a first year for us because we have a second generation fellow. Dr. Sandeep Kiswani was our 2011 fellow, and this year the awardee will actually comes from his lab. So this is our second generational fellowship baby. So at that point, I will turn this over to Dr. Matt Cooper. Just push down on the arrow, please. There you go.
All right. Good afternoon. Thank you for joining us this afternoon. I'm going to take the liberty of taking us all on a short journey, and I think you'll understand why when we come full circle, because I'm going to ask you to join a movement. At 3M, we recognize the importance of science and use it every day to improve the lives of people around the world. But we wondered what the knowledge about science was, what the impression about science, what about the trust in science in the rest of the world? Did it mirror and echo what we thought? So we looked to see whether there had been any recent surveys about such information, and there weren't. So we commissioned a survey, global study of 14 countries, of over 14,000 respondents, to ask them what their thoughts and feelings were about science. And this seems to be a great audience to share that with. The results are divided among three categories: the image of science, the impact of science, and the expectations of science. And around the world, people were fascinated with science, but quite skeptical. They didn't necessarily trust, and they doubted the results. People appreciated science, but they often took it for granted, particularly in developed countries. People have high hopes for science, as you'll see, but there are major impediments in various parts of the world to actually delivering. And it gets a little bit more complicated if you look more deeply. Nearly 40% of people say that if science didn't exist, their everyday lives wouldn't be any different. And about 1/3, in addition, are skeptical about science and its value. There is trust towards science, however, and again, to a greater degree in the developing countries as opposed to the developed countries which take it for granted. Overall knowledge about science is quite disparate. 86% of respondents, over 14,000 people globally, knew little or nothing about science. And again, the disparities are deeper. That is, the knowledge about science was lower in women and lower in adults over the age of 51, the baby boomers. So again, focuses on subpopulations, if you will, for education and assistance. And that becomes very important because when we think about our population now and population estimates, we're at 7 billion now. By 2050, the estimate is we'll be at 9 billion people globally. That's another 2 billion in 30 years. We are going to become even more dependent on science to achieve that because 70% of those people will be living in cities. That means we're dependent on the next generations to develop and participate in science. But with all this skepticism, with the lack of trust in science, with the lack of knowledge about science, how do we foster that participation in our children, in our young generations and subsequent generations? How do we help students understand that they can truly impact the world by being involved in science, that there are many different ways of participating, and we can do that through opportunities in education and equipment and supplies to help them learn and to help them be stimulated to pursue careers in science. The good news in terms of those generations are millennials are more likely to believe in science. In fact, if you ask millennials, they believe that in their lifetime, flying cars, undersea living, and teleportation will be delivered by that science. But again, it's more complicated because among those millennials, 36% of them, over a third, are skeptical in addition. So there's much work to be done. The good news is that the majority of respondents believe that innovation is tied to science and that the best days of science are still to come. If you ask adults over age 51, they believe that science will cure cancer, among other things, in their lifetime. So remember I told you that as we came full circle, I was going to try to recruit you. I'm going to try to recruit you to become champions of science, or scientific champions, to help 3M provoke a global conversation about the value, the importance, and the impact of science, about teaching about science, about learning about the impact that it delivers, to connect the dots between investing in science and teaching in science and fostering and mentoring people to participate in science and how that affects everyday life. So it's my pleasure this year to once again join with the Wound Healing Foundation on behest of 3M. This is now our 20th consecutive year for funding the 3M Fellowship Award. The intent of the award is to stimulate cutting-edge scientific research in wound healing and to support outstanding young scientists. And I'd ask Dr. Tam to come up now to announce this year's awardee.
Thank you, Matt. This year's fellow is Dr. Swathi Balaji from Baylor College of Medicine and Texas Children's Hospital in Houston, Texas. Dr. Balaji's research proposal explored the role of exosomes in the pathogenesis of fibroblast functional diversity in cutaneous scarring and fibrosis. We look forward to hearing about your research results next year in San Antonio, Texas. Congratulations, Dr. Balaji. And now for this year's fellowship lecture, Dr. Veronica Haywood from the University of Illinois in Chicago, Illinois. Thanks. Dr. Haywood will present her 2017 fellowship research entitled Enzymatic Glycosylation During Wound Healing. For those of you who have the program guide, her abstract is on page 62. And without further ado, please welcome Dr. Veronica Haywood.
I would first like to start by thanking the Wound Healing Foundation as well as 3M for the funding in order to help complete this project. Without it, it could not have been done. I would also like to thank the Wound Healing Society as well as SAWC for allowing me to speak today. And then I'll be talking to you all today about my project on enzymatic protein glycosylation during normal and diabetic wound healing. And I'm very excited about this because there's not much research so far looking at differential glycosylation outside of that related to cancer. And I have no conflicts of interest to disclose or any financial disclosures as well. So today we'll be talking about the regulation and potential roles of glycosylation and glycosylation processing enzymes during wound healing. We'll talk about the differences in the regulation of glycosylation-related genes and or glycans during each phase of wound healing in diabetic and non-diabetic skin. And we'll also be reviewing the diagnostic potential of using glycosylation-related gene and glycan expression in order to evaluate wound healing. And that seems to be the theme of the meeting this year, is to try to find biomarkers in order to help to predict whether or not wounds will heal successfully. So first I'm going to talk to you all just a little bit about why it's important and its important relationship to diabetes. We know that diabetes is an increasing epidemic impacting approximately 9% of the population, with up to 25% of those with diabetes developing lower extremity ulcers over the course of their lifetime, and up to 30% of those with lower extremity ulcers ending in a lower extremity amputation. These ulcers are highlighted by increased neurogenic inflammation, increased epithelial hyperplasia, impaired neutrophil migration, phagocytosis, and adhesion, immunosuppression and increased susceptibility to multi-drug resistant organisms, increased extracellular cross-linking, which is typically observed in basement membrane proteins like laminin, fibronectin, and type 4 collagen. And we know that this is associated with glycation, which I'm sure a lot of you all are familiar with, is a non-enzymatic process where sugars spontaneously bind onto lipids, proteins, and nucleic acids to create protein adducts and also cross-links. And these changes in basement membrane can alter the ability of endothelial cells to migrate and also to promote angiogenesis. This is not what I'm talking about today. This is very different from what I'm talking about. Today I'll be talking about glycosylation, which is an enzymatic process. It's a normal process that's required in order to help proteins fold, among other things. And during this process, there's over 700 different enzymes that are involved in glycosylation. And here you can see an example where a sugar nucleotide delivers the monosaccharide to the region, and then the glycosyl transferase synthesizes the bond in between each monosaccharide, and it can also synthesize the bond in between the proteins and the lipids that these glycan structures are added onto. It involves a number of different monosaccharides, including fucose and sialic acid, which are important terminally located carbohydrate structures that regulate cell signaling. And it's also important because there's over 7,000 different glycan structures that help to increase the diversity of our proteins and cell signaling within our bodies in all mammals. Converse to glycosyl transferases are glycosyl hydrolases, and these act to catalyze the removal of the carbohydrates from glycans and also to remove glycan structures from lipids and proteins. So some of the functions of glycosylation include its ability to synthesize the glycoproteins and glycolipids that are responsible for energy balance and the regulation of ATP. It's important in cell signaling during both the innate as well as the adaptive immune response. It's important in epidermal homeostasis, and it's also important for synthesizing the proteins that constitute the extracellular matrix, and this includes structural glycoproteins like collagens and proteoglycans. It also includes extracellular matrix affiliated proteins like lectins and mucins, as well as extracellular matrix regulators, which include those glycosyl hydrolases that remove the glycan structure from the proteins. And it's also important for growth factor activity and binding, as well as a number of receptors that are related to a number of the secreted factors, including growth factors. Some of the functions of glycosylation include protein folding, which I previously mentioned, and as a part of this, different glycan structures are added onto the protein in order to signal whether or not it's adequately glycosylated, as well as whether it'll adequately fold. And if it's not adequately folding or adequately glycosylated, then these proteins are signaled to the endoplasmic reticulum to be degraded if the glycosylation cannot be reversed or improved over time. It's also important in protein secretion. It mediates protein transit and selective targeting. It's important for providing ligands for lectin chaperones. It regulates cytosolic and nuclear functions in addition to regulating extracellular matrix interactions. It's important for immune surveillance, hormone actions, as well as it's being significantly implicated in tumor metastasis with a number of glycosylation-related gene expression data, as well as a lot of glycomics data being currently available that shows that there's differences in tumor invasiveness as well as tumor metastasis based off of the different glycans that are present. There are multiple different types of glycosylation. However, the ones that I'll primarily be focusing on are N-link glycosylation and O-link glycosylation. N-link glycosylation involves the addition of an amide linkage to an asparagine residue. And here you can see that there is a number of different steps involved in the pathway to N-link glycosylation. There are 17 different steps, which you can see listed here. And within these steps, there's 28 different enzymes that can synthesize the building of the glycan structure, which you see, that is then later added onto a nascent protein. And this is done via the oligosaccharide transferase complex. And this includes one specific enzyme known as DDOST, which is the dolichol diphosphooligosaccharide transferase, which I'll talk about a little bit more later, but all of these proteins are needed in order to stabilize this complex in order to add the glycan structure to the protein and have successful N-link glycosylation. O-link glycosylation involves a glycosidic linkage to a serine, threonine, hydroxylysine, or a glycogenin. There's different types of O-link glycosylation, and each has multiple different pathways and numbers of enzymes that are involved in regulating those particular pathways. One example of this are those that contain an N-acetylgalactosamine core, which is typically observed in mucins, and there are 7 different core structures that can be created, one of which you can see in the example below. There's also non-N-acetylgalactosamine core O-link glycosylation, or those that are non-mucins, and these are important for regulating the function in Notch, as well as regulating the activity of dystroglycan, as well as collagen formation. And then there's a third type of O-link glycosylation known as those having an N-acetylglucosamine core bound to a serine or threonine, and I'm going to discuss this a little bit more in detail later. So there's increasing evidence that there's altered glycosylation in diabetes. However, we haven't really looked at how it's impacting diabetic wound healing. It's been previously established that a number of the pathways that are responsible for mediating glycosylation can actually lead to increases in insulin resistance and the progression of the development of diabetes. This includes the role of advanced glycation end products in binding regulators of N-link glycosylation. So as I previously mentioned, the enzyme DDOST is responsible for helping to add the glycan structure onto proteins during N-link glycosylation. And this particular protein you may also know as age receptor 1. So as you can imagine, when ages bind to this particular receptor, it can interfere with its ability to stabilize this complex, which can alter overall N-link glycosylation. Additionally, we know that increases in glucose can lead to increased amino sugar and nucleotide metabolism, which then causes an increase in the formation of uridine diphospho N-acetylglucosamine, and increased OGT, which is the glycosyl transferase that synthesizes the addition of O-glycanac onto these proteins. This causes an inhibition in AKT, or it can also inhibit FOXO1 signaling, ultimately leading to increased gluconeogenic gene expression. It can also inhibit glucose transporter 4 translocation to the plasma membrane, thus decreasing uptake of glucose, decreasing glycogen synthase, and decreasing overall glycogen production. And you can imagine how changes in this glycogen production will overall impact the metabolic pathways. O-glycination is important because it affects a number of different functional classifications that are required for wound healing, and this includes transcriptional regulation, as NF-kappa B can be oglycinated, structural proteins like E-cadherin can be oglycinated, metabolic proteins including GAPDH as well as glycogen synthase is impacted, as well as a number of stress-related proteins. Another way that increased glucose can influence glycosylation is related to that of ceramide production, which is a type of sphingolipid that can be glycosylated, and it has a number of different functions in its non-glycosylated form as well as its glycosylated form. But during increased glucose production, there's a dysregulation of ceramides, which causes, again, decreased phosphorylation of AKT, inhibiting GLUT4 translocation to the plasma membrane, which ultimately leads in a decrease in glycogen production. And so you can see that there's kind of a feedback loop occurring where you have changes in glycosylation-regulated substrates that can then impact overall glycogen synthesis and then impact overall metabolism. Additionally, we know that there is a specific type of glycosylated ceramide known as GM3, which is a type A ganglioside that is previously been established to have significant impacts in its regulation of the IGF-1 receptor, and its upregulation has been shown to decrease keratinocyte migration. So as a review, when there's increased glucose and increased age formation, we know that this can impact N-linked glycosylation. And what I didn't mention is that changes in N-linked glycosylation has been shown to affect VEGF receptors, the fibroblast growth factor receptor, as well as TGF-beta receptor activity. Increased glucose alters sugar nucleotide metabolism, O-linked glycosylation. And what I didn't mention is that oglycination of proteins has an interplay with serine and threonine phosphorylation, and it kind of works together in order to fine-tune signal transduction. Another way is it can influence lipid metabolism, which then alters glycogen synthase, ultimately leading to metabolic dysfunction and increased high glucose conditions again. So since we know so much about the influence of high glucose on glycosylation and diabetes on glycosylation, I wanted to know if there was potentially any impact of glycosylation on diabetic wound healing and whether or not it led to impairments potentially in diabetic wound healing. And so the first part of my project is just very descriptive in explaining the differences in glycosylation-related gene expression, as well as the glycomics profiling that occurs throughout wound healing. So we utilize the leptin receptor deficient DB/DB mouse as our model for type 2 diabetes. And we induced 4 or 5 millimeter wounds. And we observed healing at baseline as well as over the course of wound healing, observing day 3 as the inflammatory phase of wound healing and day 10 as the proliferative phase of wound healing when we have re-epithelialization to occur in normal tissues. We also validated our model by demonstrating that there was a significant reduction in wound closure in the diabetic condition as compared to the wild type condition, with there being a significant increase in healing noted in the wild type condition at day 3, and there was really no change in the diabetic condition at this time. However, by day 10, the diabetic condition was able to demonstrate its ability to heal. However, it still did not catch up to that of the wild type condition. We also confirmed that this was a good model for type 2 diabetes by demonstrating that the DB/DB mice had elevated blood glucose as well as elevated body weights, which is typically what is observed in our patients. We then took our skin samples that were harvested at day 0 and 3, and we had them analyzed via high-throughput QRT-PCR screening platform, and we looked at the expression of 112 different genes using a sample size of 4, and then we looked at the glycomics profiling of this tissue as well, but we also wanted to look to see what was happening during the early inflammatory phase of healing, so we added day 1, and then we also added day 21 in order to examine the remodeling and resolution phase of wound healing as well. And during the glycomics profiling, we not only looked at N-linked and O-linked glycosylation, but we also looked at modifications to glycosylation, which included changes in sulfation of O-linked glycans. So during our analysis, we found that there were 38 different genes that were differentially regulated throughout wound healing between the diabetic and non-diabetic condition. These genes were found to cluster into 5 different clusters. The first cluster were those that were upregulated in the wild-type condition relative to the diabetic condition at baseline. The second were those that were upregulated in the diabetic condition at baseline. The third were those that were upregulated in the wild-type condition during the proliferative phase of wound healing on day 10. The next one were those that were upregulated in the diabetic condition at day 10. And then the last cluster were those that were upregulated in the diabetic condition during both the inflammatory as well as the proliferative phases of wound healing at days 3 and 10. So now I'm going to break up each phase of wound healing and discuss the changes that were observed during both the gene expression data as well as the glycomics profiling. So during the first 2 clusters, we found that there were 10 different genes that were differentially regulated at baseline between the diabetic and the non-diabetic condition. A number of these genes were found to be related to glycosphingolipid biosynthesis as well as ceramide sulfation. Overall, it was found that there was a downregulation of these genes in the diabetic condition as compared to the wild-type condition, and this included genes like GM2 synthase as well as enzymes that involve the sulfation of ceramides and 2 fucosyl transferases, which are important in the binding of ligands to these sphingolipids. Some of the functional implications that can occur due to changes in the glycosylation of sphingolipids and during ceramide biosynthesis include those that are related to epidermal homeostasis. Here you can see we have an illustration of normal skin, and here is normally glycosylated skin. So the stratum granulosum requires glycosylated sphingolipids to be present, and as they transition towards the formation of the stratum corneum, they lose their different glycans, and this helps to regulate the tight junction of the hydrolipid barrier. However, when there's impaired glycosylation in the stratum granulosum of these sphingolipids, then what happens is it changes the distance in between the different sphingolipids, which causes them to be more loosely bound, and therefore they don't create that hydrolipid barrier once they reach the stratum corneum during cell turnover. When this occurs, then there can also be breakdown of sphingomyelin and lysosomal conversion of sphingomyelin into ceramides, and increased lysosomal accumulation of ceramides then leads to activation of caspase 3 and increases epidermal cell apoptosis. And so then you have a further reduction of the epidermal barrier, and this allows for increased microbial invasion as well as increased transepidermal water loss. During the glycomics profiling of the intact skin, we found something quite interesting, and I will just kind of note that the glycomics profiling is our preliminary data right now. We have an N of 1, but the replicates are currently being processed. And so the glycomics profiling that I'll discuss with you all today is kind of just the profiles that already correspond with changes that we observe in the diabetic condition as it relates to like the functional changes in diabetes. So in the baseline condition, we saw overall that there appears to be an increase in end-linked glycan structures in the wild-type condition in black, respective to the diabetic condition, which is in gray. Here we found that the glycans that you can see is illustrated by having different mass spectrum mass to charge ratios in the x-axis, that there's more end-linked glycans that contain this diamond-shaped monosaccharide known as a sialic acid. And this also occurs in the second peak here. However, in the few peaks that exist that are higher in the diabetic condition relative to the wild-type condition, these don't contain sialic acids. And so overall, we see that there is a decrease in the sialylation of glycans in the diabetic condition as compared to the wild-type condition. And this was further confirmed when we looked at all of the different glycans that were found in both the N-linked glycan profiling as well as the O-linked glycan profiling. There was a decrease in the sialylation of glycans in the diabetic condition as compared to the wild-type condition. And this was further confirmed when we looked at all of the different glycans that were found in both the N-linked glycan profiling as well as the O-linked glycan profiling. There was a decrease in and sialylation in both N and O-linked glycans in the diabetic condition. This is important because selectins such as Siglec-11 and Siglec-E utilize binding to glycans that contain sialic acids in order to inhibit phagocytosis and essentially turn off the eat me signals. And so since it has such an important role in regulating the immune response, we decided to see if maybe endogenous neuraminidase were explaining why there was this hyposialylation in the diabetic condition relative to the wild-type condition. However, when we looked at cytosolic neuraminidase expression, we saw that the wild-type condition actually had higher expression of the cytosolic neuraminidase, and this would actually remove sialic acid. So this is not explaining the hyposialylation that's observed in the diabetic condition. We also looked at lysosomal neuraminidases and saw that there was no significant difference between the wild-type and diabetic condition at baseline, and so we don't expect that endogenous neuraminidase are playing a role in the hyposialylation that's observed in the diabetic condition. Some of the functional implications of hyposialylation include those related to microbial organism binding. This includes afimbril adhesions, which form on host cell surface glycoproteins and glycans bearing galactose residues. And it's also important in those that have pili and fimbriae, as these can release neuraminidases that cleave the sialic acid residues, exposing galactose residues and allowing their fimbriae and pili to extend and bind onto our cells. Additionally, after these microbial organisms release their neuraminidase, they frequently evade our immune system by moving on, while in the meantime, our immune system is now decreasing the binding between Siglec-11 and Siglec-E, which is our inhibition signaling, and instead this has the galactose residues that can then bind onto galectins and, for instance, macrophages, which then activates phagocytosis. So essentially, when there's less sialylation present, then you have increases in phagocytosis and increased immune response. And when there's more sialylation present, then you have a halting of that immune response. It's also been previously shown that in diseases such as G&E myopathy, whereby there's a hyposialylation occurring, that this leads to increases in protein aggradation and protein misfolding. This causes increased endoplasmic reticulum stress, leading to dysfunctional autophagy. It also impairs oxidative stress, which leads to dysfunction in proteasome as well as the lysosome, which is something that has been correlated with diabetes. Now, during the inflammatory phase of wound healing, we saw that there were a number of proteins that were differentially regulated during this time. However, a number of them also clustered with genes that were differentially regulated during the proliferative phase as well, and so I'll kind of split these in half. Among the genes that were differentially regulated in the inflammatory phase of wound healing, we saw that a number of chitinase-like genes were upregulated in the diabetic condition during the inflammatory phase of healing relative to the wild-type condition. This included chitinase-3-like 1, also known as YKL40, as well as chitinase-3-like 3, known as YM1. And we know both of these to be important in macrophage function, particularly with the alternative activation of M2 macrophages, which through multiple different types of pathways, including the interleukin-13-induced allergy pathway, it can alter chemokine production as well as TGF-beta production. We also know that this chitinase-like gene can bind to heparan sulfate on syndecan, which then changes the phosphorylation of FAC, which activates PI3K and ERK-mediated pathways, increasing cell migration. And particularly, this gene has been responsible for having hyperproliferation of epithelial cells, as well as increased tumor angiogenesis in the disorganized angiogenic response in cancer. We also saw that there was an increase in a unique type of glycosyl transferase that is responsible for polysialylation. And polysialylation is a conserved type of glycosylation. Here we saw that it was upregulated at day 3 in the diabetic condition, and it persisted through day 10. Polysialic acids are essentially the addition of multiple sialic acids onto a glycan chain up to 100 sialic acids in length. This is a very conserved type of glycosylation that's typically only observed in development and occurs on very few proteins, typically adhesion molecules, including NCAM, SYNCAM, podocalaxin, CD36, NRP2, and E-selectin ligand, all of which have been shown to have importance in the wound healing process. Now, I'm going to show you the glycomics profiling that correlated with this expression, and that was related to the expression of tri- and tetrasialylated N-linked glycans. And even though this doesn't represent polysialic acid, this is representative of oligosialic acids, and most likely we would have observed polysialic acid in these glycans as well. However, polysialic acid is very labile structure that is significantly altered by mild changes in pH, and so we expect that the longer chains of the sialic acid maybe fell off during the glycomics profiling, and only the tri- and tetrasialylated N-linked glycans remain. As I mentioned, polysialic acid is really only observed on adhesion molecules, and it acts as an anti-adhesion molecule. So, for instance, it prevents NCAM1 from binding to the VEGF receptor, which alters proliferation, apoptosis, and differentiation. And it also impairs the ability of NCAM to bind to other unknown receptors that are responsible for cell motility. So when there's increased polysialic acid present, we typically see that there's increased motility present in these cells. Some of the glycomics profiling that was observed during the inflammatory phase of healing was interesting in its relationship to what was observed in the intact skin, in that we saw at day 0 previously that there was an increase in sialylated glycans present in the wild-type condition relative to the diabetic condition, and this correlated with the decrease in the galactose-bearing residues, which we would expect because we know that the presence of these sialic acids kind of halts the immune response. However, at day 1, we saw the inverse occurring between the diabetic and the non-diabetic condition, whereby the diabetic condition all of a sudden had more sialylated N-glycans as compared to the wild-type condition with less galactose-bearing residues. And this kind of fits in line with the thought that there's an immunosuppressive response observed in diabetic wound healing, whereby the inflammatory response undergoes late activation and then has persistent activation afterwards when it should be resolving. During the proliferative phase of wound healing, we saw that there were a lot of different genes that were differentially regulated between the wild-type and diabetic conditions. In the 2 clusters that were solely at day 10, there were 15 differentially regulated genes in addition to the 13 that were differentially regulated in the cluster that was associated with the inflammatory phase. Here we saw that there were a number of genes related to glycan sulfation that were upregulated in the diabetic condition as compared to the wild-type condition. And this included a precursor for 3-phosphoadenosine 5-phosphosulfate, which basically synthesizes all of the sulfates that the sulfotransferases will pass on to glycans as well as the proteins. We also saw that there was upregulation of protein tyrosine sulfotransferase, which in particularly is known for sulfating P-selectin ligand. Glycomics profiling during this time revealed that there was also increased O-link sulfation in the diabetic condition as compared to the wild-type condition at baseline and throughout the entire course of wound healing. Some of the functional implications that could potentially occur due to changes in sulfation are related to that of selectins. We know that E-selectin ligand, which you can see here, requires a sial-Lewis X, but so does the ligand for P-selectin as well as L-selectin. The difference that primarily exists in these is that P-selectin is tyrosine sulfated, and interestingly, the P-selectin ligand is tyrosine sulfated, and interestingly, this particular ligand is the only ligand that can bind all 3 selectins, and so you can imagine that this could potentially have some impact on neutrophil chemotaxis. We also saw that there were a number of differentially regulated genes related to glycosamine-glycan regulation, as well as the sulfation of glycosamine-glycans. In particular, we saw that there was an increase in the regulation of heparin sulfate sulfotransferases, chondroitin sulfotransferases. And then interestingly, we also saw that a number of the genes responsible for glycosyl-Glycan degradation were also upregulated in the diabetic condition compared to the wild-type condition, with beta-glucuronidase impacting hyaluronan, chondroitin sulfate, dermatin sulfate, and heparin sulfate, hyaluronidase impacting chondroitin sulfate, dermatin sulfate, galactose-6 sulfate sulfatase impacting chondroitin sulfate, keratin sulfate, N-acetylglucosamine 6 sulfatase impacts heparin sulfate and keratin sulfate. Extracellular sulfatase impacts heparin sulfate at the same site as the GNS enzyme. And then the only enzyme that was found to be upregulated in the wild-type condition relative to the diabetic condition was beta-galactosidase. And as you can see, that this could potentially be preserving the core of the keratin sulfate structure, while the upregulation of the other sulfatases and glycosyl hydrolases are causing a decrease in the sulfation of these glycosyl-Glycans. And you can imagine how that could significantly impact growth factor binding and activity. A number of these glycosyl hydrolases and sulfatases were found to be located within the lysosome. And so one of the things that we're interested in looking into is whether or not the actual lysosomal expression of these genes is having an impact on wound healing, or if these genes could be impacting the lysosome, which is then impacting wound healing. During the resolution phase of wound healing, we only had the glycomics profiling data for this time point. And so here you can see, interestingly, when you look at the difference between day 0 and day 21 during the resolution phase of healing, the diabetic condition at day 0 mimics the diabetic condition and the wild-type condition at day 21. So essentially, intact diabetic skin looks a lot like wounded normal skin. Now I'll talk to you all about some of the comparisons that we made in differential glycosylation related to other models of wound healing, including the cutaneous versus the oral mucosal model. And I'll just note that I'm just drawing correlations between the models and not identifying these as definitive biomarkers, but further studies need to be done in order to confirm their role in wound healing. So as you all know, the tongue heals more advantageously as compared to the skin. It achieves closure sooner than in the skin. It also has decreased inflammatory cytokines, decreased neutrophils, decreased macrophages, as well as more refined angiogenesis. As you can imagine, if we were to overlay the diabetic condition over this in order to see what happens during diabetic skin wound healing relative to the oral mucosa in the skin, we would expect a dramatic response, which is over-dramatized here, but you see a delay as well as a heightened inflammatory cytokine production, heightened and delayed neutrophils, macrophages, and then decreased angiogenesis occurring during the earlier time points with peak dysregulation of angiogenesis during the latter time points. And then, of course, there'd be delayed wound closure, which we previously showed. In both of these models, we saw, interestingly, that the conserved polysialyltransferase ST8-SIA4 was upregulated in both conditions of impaired wound healing. So it was upregulated in the diabetic wounds during the inflammatory phase of wound healing, and it was also upregulated in the skin relative to the tongue during the inflammatory phase of wound healing. And as I previously mentioned, the skin has a lot more inflammation as compared to the tongue, just like the diabetic condition has more inflammation than the normal condition. We also saw that a lot of these genes were related to glycosyl-Glycan degradation, specifically the genes that are responsible for hyaluronan degradation, chondroitin sulfate degradation, terminal heparin sulfate degradation, specifically those related to the sulfation of heparin sulfate, as well as keratin sulfate sulfatases, including GAL-NS and GNS. Overall, we saw that there were a number of different proteins that were differentially regulated in both models of healing. Some of those, like I mentioned, included the polysialylation transferase. Another one included the chitinase-like enzymes, in addition to a number of others, some that are listed here and others that we haven't listed or spoken about as well. So overall, I was able to find that, yes, there's changes in N-linked as well as O-linked glycosylation during diabetes, and specifically during diabetic wound healing. Specifically, there's differences in the regulation of sialylation, and this potentially is leading to some of the differences that we observe clinically in oxidative stress response, immune regulation, as well as microbial invasiveness. We also saw that there was changes in the modification of glycans during diabetic and non-diabetic skin wound healing. And these differences in sulfation, these included differences in sulfation, which can have an impact on selectin regulation and the immune system regulation, specifically with neutrophil chemotaxis. It can also impact extracellular matrix glycosylation. And overall, we see that glycosylation likely does play a role in the differential healing response. And so the NIH is currently looking to promote increased studies in glycomics, and so I encourage you all to utilize glycomics as another omics tool for biomarker discovery. I would like to thank Dr. DiPietro for allowing me to kind of go onto this side field of discovery for looking at glycomic profiling in different wounding models. I'd also like to thank Lynn and everybody else in the lab for helping to assist me. Dr. Karen Colley, who is a glycobiologist in the College of Medicine, who's helped a lot, as well as those at the Complex Carbohydrate Research Center, who helped with the high-throughput QRT-PCR screening platform, Dr. DLEU, who's doing the glycomics profiling, and then also Dr. Kelly Archer at Ohio State, who assisted with the statistical analysis of the high-throughput gene expression data. And then also to the Wound Healing Foundation and 3M, as well as our other funding partners at the NIH and the NSF. Thank you.
This research is open for questions, so please come to the mics. They're at the front of each aisleway.
While people are making their way up there. Thank you for that great talk. I actually have two questions. The first one is you described glycosylation as having very broad effects on a lot of different proteins, and some of these are structural things like collagen that typically don't turn over very much. So is it known if there are long-lasting tissue memories, if you will, where these changes would persist even after, say, someone's diabetic status is improved?
Well, for certain proteins, this could have a potential lasting impact just because of their slow turnover. And we expect that even in cases where there has been resolved diabetes, the advanced glycation end products typically are still residing in the extracellular matrix proteins like collagen 4. And so we would expect that the impact of those ages would still continue to influence the process of glycosylation. Over an extended period of time, it will perhaps improve. However, you're talking about years between the time that the extracellular matrix proteins are turning over, especially in aged skin.
Thanks. Go ahead.
That was a great talk. Lots of data. So I noticed there you had the hex A/B. Looked like it was knocked out or its effect, and then you started talking about lysosome problems. Is there, before you dive into that, is there some kind of like simple staining you could do to look at cells in some other diseases where lysosomal storage problems occur that you could, compare it to before you, went down that path?
Yeah, there are, especially like muscular dystrophies, a number of those are diseases of glycosylation, and so we could look at the differences in the lysosomal distribution within the cells, their permeability, as well as the different enzymes that are found within those lysosomes to see if there's coincidence staining between the lysosome and those respective proteins. And then we can compare it between like the two conditions of the impaired glycosylation, and then also look at it in the normal and the diabetic condition in order to cross-reference them.
Great. Good luck.
I need your help. Great talk. Thank you. I learned a lot about the glycosylation. I am not quite clear what you mean when you refer to the modifications of the ceramide that then will affect the way the GLUC4 transporter goes to the membrane. Could you elaborate a little bit more so I can think?
Yeah, so in the laminar bodies within the stratum granulosum, it contains a number of glycosphingolipids, and these are important to help strengthen the interactions between the lipids within the plasma membrane of the stratum granulosum. And as those enucleate, then they are moved closer together, but then those glycans are kind of allowed to fall off at the same time, which helps to decrease the space between the different lipids, increasing the interactions between them. Ultimately, it helps the stratum granulosum to have a structurally sufficient plasma membrane, and that structurally sufficient plasma membrane then, as it's degraded, turns into the hydrolipid barrier, which is present in the stratum corneum. So if it has more of a permeable membrane as it's in the stratum granulosum, then it's not going to have that strong hydrolipid barrier once it reaches the stratum corneum.
Yeah, but the way you showed it in your picture, I'm trying to figure out which compartment really contains the ceramide that is affecting then the AKT, and which then affects the transporter.
So I think that it's probably a combination. When we look at the model of like GM3 and its increased expression, we know that there's increased apoptosis occurring, there's decreases in the cell migration that's occurring as well, and GM3 is particularly a glycosylated type of ceramide, and so potentially one of the reasons why there's differences observed in that model is because we actually want there to be more of the GM2 phenotype of these sphingolipids as opposed to the GM3 phenotype of the sphingolipids, and that will change the cell signaling. I'm not exactly sure of the interactions within that pathway, but I know that, like in diabetes, there is increased GM3 production. However, from what we show, we really want more of the GM2 phenotype to be found instead.
Thank you.
I think we should probably close it. So we're going to close the session for now, but stay put. We're going to give out a few foundation awards and then immediately transfer into the business meeting. Veronica, thank you so much. All right, so just a few quick awards. If you are called, please come forward, and Manuela will hand you your certificate, and then please go to the side, and we will take pictures outside so that we don't interrupt the society meeting that's coming up next. So if you were here at the beginning, you heard that we had our second fellowship generation of wound healing fellows, so Swathi Balaji had received the fellowship. So go ahead and come up, Swathi, and start the line, please. We'd also like to recognize Alessandra Moore because she had an incredibly innovative fellowship proposal. This was very tight. It was 0.2 of a point between the winner and the runner-up. And for that, we would like to recognize Alessandra for her contributions and her innovativeness and provide her with a travel award. So thank you very much, Alessandra. We'd also like to offer the Impact Award to Maximilian Kugelhaus for his contributions in the wonderful work that he did on EB with the patient. So please come forward. We have a lovely plaque for Dr. Kugelhaus, as well as a small honoraria. We appreciate you sharing your science with us and helping us advance not only the technical side, but also the clinical side. So thank you very, very much for joining us. And this is the Foundation Impact Award. We also had several travel awards that we gave out this year. Michelle Bach received a travel award from her high-ranking abstract submission, and she gave the oral presentation the other day, and it was just outstanding. So we were very happy to offer that to her. Unfortunately, she had to get back to class today. We also, from Ohio State, have Kanahan Singh. So if he is here, if Dr. Singh is here, please come up and join us. And again, he also gave a very good oral abstract just earlier this afternoon. And then Mariel Walraven also had an extremely high-ranking abstract submission. She's at the University of Toronto, and we would like to recognize her for her work. So if she's in here, please come forward and join us. We also like to recognize some of the sponsors and donors. Without the sponsorship and donations from different generous individuals as well as corporations, we wouldn't be able to promote the Foundation's mission and enhance the offerings of the Society. So we'd like to actually give the Generosity and Giving Award to Phil O'Neill from Swiss American Products, because the company and Phil generously contributed over $125,000 worth of product for our Wounded Warrior Initiative, as well as an additional $1,000 in product for our tribal healthcare initiative and outreach programs that we had for indigent care clinics that specifically work in wound care. And he was unable to join us today, but we feel it's very important that we recognize our sponsors and donors and supporters. We also would like to recognize Komal Grover. She's rotating off of the board. She's been a board director for 6 years on the Foundation, and she acted as the Vice President of Operations within the Foundation for 2 years, and she was not able to join us today. We also had Brian Gamble was a board of director for the past 3 years. He's down in Florida at the university, at the VA hospital down in Orlando, and we sincerely appreciate his help and contributions in helping us in our efforts and helped lead the way through the federal campaign programs with us. We'd also like to recognize Paul Ehrlich. Paul was not able to join us today. He was a very long-lasting awards chair for the Foundation from 2006 to 2017, and 11 years is a long time to be an awards chair. So he did a fantastic job. He was also a director on the board for the past 9 years, and he has had so much support and really giving back to the wound community, always been a very strong supporter of both the Society and the Foundation, and we want to recognize his efforts. And then last of all, we would very much like to extend our certificate of appreciation to Elof Erickson, who has been on the Foundation Board of Directors for the past 2 years as an ex officio. And so you give it to Elof. So at this point, I would like to thank everybody for your attendance in the fellowship and for our award ceremony. And I'd like to turn it over to Elof for an on-time start of your Society business meeting. So thank you for coming.
Thank you, Laura, for chairing a very impressive session. And thank you also. I want to thank the Wound Healing Foundation for everything that you are doing for our members of the Wound Healing Society. I'm very impressed with your hard and compassionate work. Thank you.
Thank you.
And we are going to go a little out of the usual order here. Let's see if we can get my slides up here. Sorry? Okay, thank you. If you give us a few seconds to put up the slides, maybe you can stretch your legs in the meantime.
Can I just sit down? Okay. Jeff Gertner in the room. Tracy, come up here. Yeah. Many chairs. Yoav is not going to sit. It's his show. I'm at the tail end. So it's Jeff next to Sashwati. Right?
Oh.
We're going in order of importance. Order in importance. I'm down here. You are next to me, and then I'm at him. Where's Alan? He was just here. Hmm? Oh, Brad. Well, no, Bert.
Yeah, I know. I just got one more from the rapid fire. So here's my chance, you know? And we're still missing one.
It's all yours.
Thank you. And the changer is this one? Or?
Just give
Okay, will do. Once again, welcome everybody to the WHS business meeting. And I'm very pleased to be here with Sashwati Roy, who will be our president within the hour, and Jeff Gertner, who moves up to be the president-elect. Mariana, who has done a fantastic number of years of service to our society in a great way, and Tracy Wilgus, who has been our great secretary. I don't know for how long you have done that, 2 years. It seems like you have helped us forever. And finally, Bram Schroot, who has been our, do we call you Chief Financial Officer? And Treasurer. But he has really functioned as a Chief Financial Officer and done it in a fantastic way, to the point where our finances are in better order than ever. So thank you, all of you. We are going to go a little bit out of our usual order because one of the people we are honoring has a time conflict, so bear with us. It won't matter in the total picture. And so we are going to start with the Wound Healing Society Awards. And it gives me great pleasure to present the Lifetime Achievement Award to Big Bill Eaglestein. He has, I'm going to say a few words and then ask you to come up, Bill. He has, I think of everybody present here at this meeting today, he has probably contributed more to wound healing than everybody. And for instance, he has written a large number of great papers for over 38 years. He started out after medical school in the military, did his training in dermatology in Miami, quickly advanced through the ranks and became the chair of dermatology in Pittsburgh, then moved back to the South, where he has developed one of the best, if not the best, dermatology and wound research programs in the country. He has been recognized widely and received a huge number of awards and great accolades. And I am very pleased that we can add the WHS Lifetime Achievement Award to all the other ones Bill, this is just great. Please.
Thank you, Elif, and thank the Society for this outstanding award. By good fortune, my wife is in town, and of course I want to recognize her role in my career. But beyond that, I would like to just say that as everyone in our role, you've had lots of people who've helped you in your careers, and I have as well. Some are even here in the room. Oscar Alvarez was one early collaborator. But I wanted only, or primarily to mention and think of Patricia Mertz, who I hope many of you knew and know, or did know. She passed away a few years ago, but for me she was a most important element, a tremendous investigator, experimentalist, and I worked with her for 40 years, so I feel very much that I'm indebted to her for this award and other successes in my career. Thanks.
Thank you again, Bill, for everything you have done for wound healing and our patients. And I'm going to now let the awards committee, do we have Praveen and Irina here to continue the presentations of the awards. Praveen and Irina are chairing the awards committee, and that may sound like an easy job, but I have been very impressed with their hard work and objectivity.
Right. We have a set of awards that we'd like to give out. All the nominees for these awards were vetted by the awards committee, and they were voted on to ensure there are no conflicts of interest. The most prestigious award that we give out is the Anita Roberts Award. Every one of you know who Anita Roberts is, one of the co-founding members of the society. This is given to the best young investigator who's published a first author paper in our journal, Wound Repair and Regeneration. And this year it's awarded to George Glinos for optical coherence tomography for assessment of epithelialization in a human ex vivo wound model. George.
Thank you.
Congratulations.
The next series of awards are going to be announced here, but they're going to be given physically at the poster gala. As the WHF, Laura pointed out, this year we had a bonus of travel awards. There were 4 from the Wound Healing Society, WHS, 3 from Wound Healing Foundation, and 4 from the Wiley publishers. These are the names. I'll recognize you here, but please be present at the poster session where you will get your actual award. Mohammed Ashrafi, Michelle Bach, Ayan Biswas, Clark Bonham, John Luo, Vikram Mukherjee, Kanaya Singh, Mithun Sinha, Christopher Turner, and Marielle Walleran. Please give her a big hand. They will all receive travel support in the form of a check that WHS will give you. The next award recognizes excellence in translational regenerative research as applicable to wound healing. This award specifically recognizes scientific work that fosters the development of cell-based and other therapies in wound healing and regenerative medicine towards clinical applicability. The winners this year are Dr. Subhadeep Ghatak and Dr. Leslie Gold. The next award recognizes a junior faculty in the field of wound healing research and enables them to attend this annual meeting. This year, the recipient is Yuan Zhao. We have a group of members who are from the industry, and to promote further interaction between the academic and the industry partners, we recognize the best work that is presented as a poster. There were 8 finalists this year, and the winner this year that was judged this morning is Dr. Amitabh Das for his work on a collagenase preparation for wound beds. We also congratulate all the other presenters who presented their interesting and innovative work, and encourage the industry members to participate actively next year. The biggest award that the Wound Healing Society gives is to recognize the junior investigators and promote their interaction in the forum further. This year we had 7 finalists, and as noted here, the winner this year is Ivan Joik, who was given the Young Investigator, and we will be presenting his work at the ETRS meeting. The other 3 finalists, Dr. Michael Hu, Georgios Therakarides, and Amitabh Das, were also highly scored and are recognized as finalists. At the behest of the Communication Committee Chair, Mo, who's in the front right there, we've been trying to promote our social network and our social networking skills. This year, we initiated an award to recognize folks who are interested in enhancing communication and specifically highlighting the meeting experience at Wound Healing Society. This year, the award, the first time is given out, goes to Dr. Lisa Tucker Kellogg. She will receive a 1-year free membership at the Wound Healing Society.
Thank you once again, Praveen and Irina, for your great work. And I will say a few words about this year as president. It has been very interesting and very exciting, and I've also learned a lot. And I am pleased and proud that you have let me be your president for a year. We have done a few things. We changed the management to PRRI, and we had high expectations, and they have performed way beyond our expectations. And I would particularly mention the president, Aurélie Alger, and also Katie Levitt, who has done the grunt work since we started, and Crystal Beatrice, who is the manager on our side of this meeting. Katie and Crystal, could you please stand up, and we'll give you a big hand. Great work and way beyond our expectations. Thank you. We have also started to work on something that we call the Best Practice Project, and we wanted to be just that, to be able to give answer to what's the best practice in clinical care and in research. We are not off the ground yet, but we are working on the algorithms, And when we get the algorithms to a state that we're happy with, we will start putting up templates. So stay tuned. There should be a good development of that during this year. Once we get going, we're also going to call upon you, the experts in various areas, to help us. How are we going to do it? Well, I would describe it as a combination of the OED, the Oxford English Dictionary methodology, and the Wikipedia, because it will be a big project, and we hope to keep it alive as we go along. Two branches to this, one for practitioners, one for patients, and we want it to be free with easy access and be one subsection of our website, all with the goal of better communicating our knowledge and more point out the relevance of the Wound Healing Society to the wound healing community at large. We have also started to prepare for contract negotiations for future meetings. In the past, we have often been caught short of time. We are trying to seriously be able to present our board with various alternatives to evaluate in due time before decision making. And before going on, I would also like to mention two things. It won't be a speech. I'll just communicate two impressions to you. And one is that during this year, I have become very impressed with the knowledge, capabilities, focus, and compassion of the members of our society. We should be proud members of this society because there are a large number of very capable and dedicated people in our group. And it has been a lot of fun to work with you. Of course, most of my interactions have been with the executive committee, the board, and the chairs of the committee, but uniformly, I have been very positively impressed with how many superbly talented people we have among our ranks. The second impression is more of one of the meeting and our presentation. I should probably mention that in my early research life, I had a series of R01s with the NIH. Now I do trauma research completely funded by the DoD, and that has probably made me think in this direction. I would like to make a plea for thinking of our patients with wounds. You'll say that's obvious, but when you come to the various sessions at our meetings, sometimes you are tempted to think that this is a session of general skin pathophysiology, and sometimes it is. I would just like to say that even if it's extremely important that you please the NIH and get the next grant, I also want you to perhaps once a week think of our patients with wounds, and just like, for instance, the DoD expects that you are able to plot a path from your research to something benefiting patients in less than 5 years. They prefer 3 years. Maybe we all should try every so often to plot a path from our basic research to direct practical patient benefit within 5 to 10 years. Of course, many times we won't be able to achieve that, but I think the mental exercise of remembering that we are here, we are the Wound Healing Society, and we are here to heal the wounds of patients first, and please the NIH second. I don't think it decreases any of what we do for the NIH. As a matter of fact, we may help them focus more on what they are supposed to focus on. But particularly in this group, please don't forget the patient with wounds. I'm going to move on, and it would be very nice if we could get each one of these great people to give a report, but then we would completely miss the poster session, and I don't think anybody wants to do that. So I'm just going to say thank you to Bram for all the things you have done, and in particular this year we have had our best financial year ever. Bram has masterminded an audit which came out very well, and we got a very clean and advantageous report. He has also masterminded the change to an investment advisor that is much more geared toward serving a society of this kind than the boilerplate type of advice that you get when you engage a large investment house. And this is in addition to the numerous weekly, sometimes daily requests for a sound advice from the treasurer that we have been asking. And Bram has not only done it in an expert fashion, he always does it with a smile. And I don't think you can ever make this man lose his good humor. Thank you, Bram.
You're welcome.